Okay, so let's start chapter 11, okay? So as you can see, chapter 11 talks about the nervous system. So when we say nervous system, what do we mean by that? Your brain, spinal cord, all the peripheral nerves, right? These are all the part of your nervous system. So what is the function of nervous system? Sending the signal? I would say the communication. So everything you have done in your whole life, like if you think about it, everything was possible because you do have a nervous system. Just imagine you didn't have a nervous system. Would you be sitting here listening to me? Just for listening to me, you need your nervous system, right? To understand, you need your nervous system. Even to sit down, you need your nervous system. Even to see, you need your nervous system. So your nervous system actually communicates all the different parts of your body for you to function properly, right? Okay, so one of the example, as you can see right here, like how is it happening? Even whenever you are thirsty and then you see, first of all, you need to understand that you are thirsty. To be able to understand that, you need your nervous system. And then when you see a glass of water, so what do you want to do? You need the nervous system to see it, to take the information in your brain, integrate that in your brain, and then take the action to grab this what water cup and actually drink it. All these different processes are actually done because you did have a what? Nervous system. So when we say the function of the nervous system is the communication, what is actually happening? So not only you take action by using your nervous system, but also what? Take the information from the environment by using the nervous system, right? Okay, and then you integrate the process and then you take action, right? So now you have neurons which take the information. We call them sensory neuron for the sensory input, and then there are some neurons which help you take the action. We call them motor output or motor neuron. And in between the sensory neuron and the motor neuron, what do you have? You have the interneuron for the integration. Does it make sense? Yeah. Okay. So now, as I have been telling you earlier, that when we think about your nervous system is made up of your brain and the spinal cord. We call it central nervous system. And then you have all these nerves which are coming out from the brain. We call it cranial nerves, and the nerves from the spine, spinal nerves, the peripheral nerves. These are all the part of the peripheral nervous system. Okay, so you have your central nervous system, which is made up of the brain and the spinal cord, and then you have your peripheral nervous system, which is made up of all the nerves from the brain and the spine. Okay, so as you can see, you have your central nervous system, which is always communicating with the what? Peripheral nervous system. So first of all, you need to think about is you have that sensory division, which is taking the information from the environment. So your sensory division is taking the information from the environment, and through the peripheral nervous system, you are sending the information to the what? Your central nervous system, and then central nervous system is sending the information through your peripheral nervous system to the motor division so that you can take the action. That taking action can be through the somatic nervous system, or it could be through the autonomic nervous system. If it is done through the somatic nervous system, usually voluntary. Like for example, all the skeletal muscles are actually working due to the what? Activity of the somatic nervous system, right? Similar to that, your autonomic nervous system. Mostly this autonomic nervous system is regulating your cardiac muscle, smooth muscle, and the gland. Like for example, I can give you some of the example of your somatic nervous system. Like for example, the example we just have seen, to take or grab a glass of water to drink, that is your somatic nervous system. But when we say autonomic nervous system, that is happening, like for example, now we are all listening, you are all listening to me, and all of a sudden you saw a tiger in front of the door. What would you do? Probably you do not even think about it, it's already inside your body, your heartbeat increases, all these things are happening. So that is happening by the what? Autonomic nervous system. So that autonomic nervous system has actually two division. One is called the sympathetic division, another one is called the parasympathetic division. So the example I just gave you, you saw a tiger on the door, so that is probably is going to activate your sympathetic division. But when we say parasympathetic division, what is happening? It's basically telling you rest and digest. Whenever you are calm and relaxed, you want to sleep, that is when your parasympathetic division is what? Working. Okay, so usually when we say sympathetic division, what do we usually mean by that? Okay, so there is a specific way to say like sympathetic division, and the parasympathetic division is actually what? Rest and digest. Okay, so now there are only two different types of the cells that you find in your nervous system. Number one is called the neuron. The neuron, which we have been talking a lot about. That's the most important cell in your nervous system. That's pretty big, and it has a lot of different parts like axon, dendrites, cell body, and all this. And there are some other helper cells, we call them neuroglia or glial cells. These are very small that surround and wrap delicate neuron, and they have various functions. So now let's start with the function of the neuroglia. We have different types of the neuroglia. First of all, we have the oligodendrocyte. So to be able to understand the oligodendrocyte and neuroglia, I actually have to draw a neuron first. So for example, if I draw a neuron, we know that in the neuron you have what? Cell body, and all these small projection, we call them what? Dendrite, right? And then you have the what? A long projection, we call it axon, right? On top of the axon, you have some neuroglia. It's almost like a sausage roll inside the what? Bread. Okay, this sausage roll is your neuron, and the bread on top is called the myelin sheath. What do we call it? Myelin sheath. This myelin sheath is made up of neuroglia. So if we were thinking about the neurons of the central nervous system, which is basically the what? Central nervous system neuron, which is your spinal cord and the brain. The neuron in your central nervous system has the myelin sheath made up of the neuroglia, which we call it oligodendrocyte. So oligodendrocyte is the cell which is what? The part of the myelin sheath of the neuron present in the central nervous system. Okay, but this myelin sheath is made up of a different type of the glial cell or neuroglia in the peripheral nervous system. We call them Schwann cell. Okay. So on top of those, there are some other cell, like for example, astrocyte. So astrocyte is also another type of cell which is largely present in your central nervous system, help form the blood-brain barrier, regulate interstitial fluid composition, provide structural support and organization to the central nervous system, and assist with the neuronal development, replicates to occupy space of the dying neurons. And then there is another type of neuroglia, we call them ependymal cell. So before I talk about the ependymal cells, I want you to understand that today in your lab you are going to dissect the brain. So when you dissect the brain, you are gonna see there are some fluid filled cavity inside your brain. So you call them ventricles. What do you call it? Ventricles. So those ventricles are lined with the ependymal cell. The ventricles are lined with the what? Ependymal cell. So the ependymal cell which lines the ventricles of the brain and central canal of the spinal cord, assist in the production and circulation of the cerebrospinal fluid. So as I was telling you, your fluid filled ventricles. So the ventricles are lined with the what? Ependymal cells. So what is the function of the ependymal cells? To circulate and produce cerebrospinal fluid. So the function of the microglial cell. So there is another type of the neuroglia that you have in the central nervous system. We call it microglial cell. So the most important function of this microglial cell is to actually do the what? Phagocytosis. Who remember what is phagocytosis? So when you have anything bad that are trying to attack you, what do you want to do? You want to swallow it. Yeah, that is the phagocytosis, right? So microglial cells are the phagocytic cells in your brain. Okay, they are trying to see if anything bad which is trying to attack your brain. It's almost like trying to make sure you are safe from the attack. Okay, so phagocytic cells then move through the central nervous system, protects the central nervous system by engulfing infectious agents and other potential harmful substances. So as I told you already about the astrocyte, these are the most abundant, versatile and highly branched of the glial cell, cling to neurons, synaptic ending and the capillaries. So what is the function? They support and brace the neuron, play a role in exchanges between the capillaries and neurons, guide migration of the young neurons, control chemical environment around the neurons, clean up leaked potassium ions and recapture and recycle neurotransmitter, influence neuronal functioning. So as you can see, a lot of astrocytes in the what? Astrocytes the most abundant in the central nervous system, neuroglia in the central nervous system. So microglial cell, I just talked about the microglial cell. What is the function of the microglial cell? One more time. Phagocytosis, right? So small ovoid cells with the thorny processes that touch and monitor neurons migrate toward the injured neurons and can transfer to phagocytize microorganisms and neuronal debris. So I think I did ask question about it in your exam. So make sure you know the function of microglia. Okay, so what about the ependymal cell? I just told you about the ependymal cell. Where are they located? In the lining of the ventricles. So what is the function of the ependymal cell? Something with the cerebrospinal fluid. What does it do? Form permeable barrier between the cerebrospinal fluid in cavities and tissue fluid bathing central nervous system cell. Okay, so as you can see, the ependymal cells, how they are lining the fluid filled cavity called ventricles. Then I told you already, if your neuron is present in the where central nervous system, and their myelin sheath is made up of the oligodendrocyte. So these are the branch cells with the fewer processes than the astrocyte. So there are some other neuroglia that is present in the peripheral nervous system. One of them is called the satellite cell. Satellite cell is almost like the astrocyte, and they are present in the peripheral nervous system and function like the astrocyte. And similar to that, in the peripheral nervous system, you have the Schwann cell. The Schwann cell is actually making up the myelin sheath of the neuron in the peripheral nervous system. So satellite cell and Schwann cells are the what? Specific for the peripheral nervous system. Okay. Your satellite cell work like astrocyte, and Schwann cells act like oligodendrocyte. Okay, as you can see in your peripheral nervous system, you have this myelin sheath which is made up of the what? Schwann cells. And these right here around the cell body, these are called the satellite cells. So what are the characteristics of the neuron? Since we know the function of the neuron is the what? Excitation and conduction of conduction and communication, right? So definitely your neuron should have the excitability, conductivity, and very important that they have extreme longevity. So when we say extreme longevity, what do we mean by that? It means that after the birth, from the birth to death, you will have those cells alive unless you damage it by any other way. Okay, for example, the stroke can damage, right? Or any other way that you can damage your what? Neuron. So if you damage your neuron after you have the stroke or damage in the brain, it's so hard to actually regenerate because your neuron do not what? Divide. Well, okay, so they are amitotic with few exceptions. So they have very high metabolic rate, requires continuous supply of the oxygen and glucose. So as you can see right here, you have a neuron which has a cell body. Inside the cell body, you have the nucleus, endoplasmic reticulum, and then the mitochondria, all the different organelles that you know. Okay. So they have a lots of rough endoplasmic reticulum. The endoplasmic reticulum of the neurons are called Nissl body. The endoplasmic reticulum of the neuron is called what? Nissl body. Okay, so that is something different. In the muscles fiber, the endoplasmic reticulum is called the what? Sarcoplasmic reticulum. But in the neuron, those endoplasmic reticulum is called what? Nissl body. So cell body, perikaryon or soma, okay. And then they have this long projection. They have some small projection. We call them dendrite. A lot of the time, these dendrites are actually what? Receptive, okay. So they take the information, receptive region. And then you have the axon right here. Axon is the one which basically does the what? Conduction, right? Impulse conducting. The place where your neuron starts from the cell body is called the axon hillock. Is called the what? Axon hillock. And on top of the axon, you have the what? Myelin sheath. It's almost like the what? Sausage roll and bread on top. That bread is called the what? Myelin sheath, and the sausage roll inside is called the what? Axon. Okay. So the myelin sheath, if they are present in the central nervous system, they are made up of the oligodendrocyte, and if they are present in the peripheral nervous system, they are made up of the Schwann cells. So in between the each bread, do you see? In between the each myelin sheath, they have this place, they have these nodes right here. Those nodes are called nodes of Ranvier. It's a French word, Ranvier. Okay. So the nodes of Ranvier. Okay. So whenever you are trying to conduct the action potential or electrical signal, what they have to do? They actually have to run the signal from what? Nodes to nodes, nodes to nodes, jump from one nodes to nodes, right? If they have myelin sheath, the conduction of the signal happens very, very quickly. Yeah. So as I have been telling you, dendrites, these small projections, receptive region of the neuron, and axons. Each neuron has one axon that starts at the cone-shaped area called the axon hillock, can be short, long, or absent, and long axons are called nerve fibers. And sometimes they do have some branching. The branchings are called axon collaterals. And distal endings are called axon terminals or terminal botons. Okay, so what is the function of the axon? First of all, I want you to understand that the cell membrane of the axon is called the axolemma, and at the end of the axon is called the axon terminal. So you are conducting the signal from the what? Axon hillock to the what? Axon terminal. So that can happen in both direction. It can happen anterograde away from the cell body in this direction, or retrograde toward the cell body in this direction. Okay. So just one more time to make sure that you understand the different parts of the neuron. The blue right here, where your neuron axon starts from the cell body, is called the axon hillock. The cell body is called the cell body, of course, and the nucleus inside the cell body, and the long projection axon, the small projection dendrites, and nucleus inside the cell body, and these are the nucleus bodies inside the cell body. So there are some terms. I did ask some questions in your exam from this slide. So make sure you understand that the same thing, same part of the different parts of the neuron, but they are called different in different system. For example, if you are thinking about the central nervous system, you do not call the same thing in the peripheral nervous system, right? For example, if you look at the clusters of the neuron cell bodies in the central nervous system, if you look at the cluster of the neuron cell bodies, they are called nuclei in your central nervous system, and they are called ganglia in the peripheral nervous system. But in both cases, what we are talking about, the clusters of the neuron cell body. And then they have the arm-like processes, like central nervous system contain both neuron cell bodies and their processes, and peripheral nervous system contains chiefly neuron processes. And these bundles of the neuron processes in central nervous systems are called the tracts, and then in the peripheral nervous system they are actually called nerve. A lot of the time you will hear about the what? Peripheral nerve. Peripheral nerve. Okay. So the tracts and the nerve. I already talked about the myelin sheath. So whitish protein lipid substance that protects and insulates the axon, increases speed of the nerve impulse. Non-myelinated fibers do not contain the myelin sheath and conduct impulses more slowly. So do you understand what they are talking about? As I told you, myelin sheath is what? They are the neuroglia on top of the axon. If you have the myelin sheath on top of the axon, you can conduct very quickly. You can conduct the signal very quickly. But if you do not have this myelin sheath, you can conduct, but the conduction is gonna be very, very slow. If they have the myelin sheath, from the nodes to nodes, signal conduction happen by jumping. So it's gonna be very, very quick. So now I already talked about all these things. So one more time, formed by the Schwann cells, wraps around axon in jelly roll fashion. One cell form one segment of the myelin sheath. An outer collar of the perinuclear cytoplasm, neurolemma, peripheral bulge containing nucleus and most of the cytoplasm. So these myelin sheath gaps are called what? Nodes of Ranvier. The myelin sheath gaps are called the what? Nodes of Ranvier. These are the gaps between the adjacent Schwann cells, sites where axon collaterals can emerge. And if they do not have the myelin sheath, we call them non-myelinated fibers, thin fibers not wrapped in the myelin and surrounded by the Schwann cells, but no coiling. Only cell may surround 15 different fibers. So as you can see, the myelin sheath around the axon. So myelination in the central nervous system. So this is a repetition of the what information. So I told you already, if you are thinking about the myelin sheath in the central nervous system, they are always made up of the oligodendrocyte. Each cell can wrap up to 60 axon at once. So one oligodendrocyte can actually wrap 60 axons at once. And myelin sheath gap is present, no outer color of the perinuclear cytoplasm, white matter regions of the brain and spinal cord with the myelinated fiber. And when we say the gray matter neuron does not have the myelinated fiber. So have you ever heard of the part of the brain which are made up of the white matter and the gray matter? Your spinal cord also has this white matter, gray matter. So when we say white matter, they are made up of the what? Myelinated axon. If they are made up of non-myelinated, they are what? Gray matter. White matter is made up of what? Myelinated, and gray matter is made up of non-myelinated axon. So now I also want you to understand that these projections that you see from the neuron can be one, two, or multiple. If your cell body has multiple projections like dendrites, axon, then you call it multipolar. And if they have two projections from the cell body, you call it bipolar. That is very rare in human body. And there is also unipolar. As you can see right here, unipolar, from the cell body only one projection is coming out, and that projection is getting branched out. So usually your what? We have three different type of the neurons, right? We have the motor neuron, interneuron, and sensory neuron. Usually your sensory neurons are made up of the unipolar and interneuron, and the motor neurons are made up of the multipolar neuron, as I told you before. So as you can see, your sensory neuron is usually made up of, look at this, so one projection. Sensory neuron is made up of the what? Sensory neuron is made up of the unipolar or multipolar? Unipolar. And your what? Motor neurons are made up of the multipolar neuron. So there are some diseases that can happen due to the nervous system disorder affecting the myelin sheath. So one of them is the multiple sclerosis, progressive demyelination of the neurons in the central nervous system, autoimmune disorder, so oligodendrocyte attacked by the immune cells, repeated inflammatory events causing scarring and permanent loss of the function. So as I told you, if you are breaking down your breaking down the what? The myelin sheath, what will happen? You are going to be very slow in processing all the information, right? Taking information, taking action, processing all the information, because neuron is the one which is actually what? Sending, taking, receiving all the signals, right? So myelin sheath is very important. If you have myelin sheath, you can do everything very efficiently and quickly, okay? But due to the multiple sclerosis, if you are actually damaging your myelin sheath, what will happen? The conduction will be very, very slow. And there is another syndrome which is called the Guillain-Barré syndrome, loss of the myelin from the peripheral nerve due to the inflammation, and muscle weakness begins in distal limbs, advances to the proximal muscles, and most function recovered with the little medical intervention. Okay, so now question for you, is the autonomic nervous system considered a sensory or a motor system? Autonomic nervous system, sensory or motor? Of course, motor system. Very good. What is the difference between the axon and dendrite? Dendrites are small projection and usually receptive. So they receive the signal. What about the axon? Axons right here. Long projection and conduct the signal, right? So what myelinates axon in the peripheral nervous system? So the myelination happen with what type of cell in the peripheral nervous system? Oligodendrocyte for the central nervous system, but for the peripheral nervous system, Schwann cell. Yeah. Which neuron has only one extension off of the cell body? Unipolar. Very good. What is the most common functional class of the neuron? Yeah, multiple. So the multipolar. Okay. So now let's come back to the resting membrane potential. So we just talked about it. That's why I was talking about it a lot. So everyone now remembers what is the resting membrane potential? So we know that in our cell, what do we have? In our cell, what do we have? In our cell, we have the sodium potassium pump. What is the function of the sodium potassium pump? Take out 3 sodium and bring in 2 potassium. When that happens, what happens? The charge outside the membrane, which is the extracellular fluid, and inside the membrane, which is the intracellular fluid, changes. So this becomes more negative. In terms of the neurons, that's usually negative 70 millivolt. When your neurons are at resting condition, they have this negative 70 millivolt. What? Negative 70 millivolt is the what? Resting membrane potential. So when I say resting membrane potential, what does that mean? Your neuron is not working, is not excited, right? To excite, for excitation, what do you have to do? This was polarized, so you have to, for excitation, you have to depolarize. For excitation, what do you have to do? You have to depolarize. So I did talk about the extracellular fluid and the intracellular fluid. So extracellular fluid has the higher concentration of sodium because of this sodium potassium pump, and then intracellular fluid has the higher concentration of potassium because of this sodium potassium pump. So potassium plays most important role in the membrane potential, 25 times more permeable to the potassium than sodium, diffuses out along the concentration gradient, thus inside is more negative. So inside is what? More negative. So two main types of the ion channel, leakage channel, always open, and gated channel, protein changes shape to open or close the channel. So remember the gated channel, that one, acetylcholine receptor bind with it and the sodium enters, that was the gated channel. But there are some channel which are leakage channel, you do not have to open it, they are kind of always open. You can always come out and go with that door, okay? So that is the leakage channel. So potassium can easily go out and go inside with the leakage channel. But for sodium, what do you have to do? You have to have the gated channel. Somebody has to open the gate, or maybe somebody has to use energy to what? Transport the sodium. So as you can see, for sodium, potassium, the gate can be closed or open. When the gate is open, sodium is coming out and potassium is going out. So when that happens, that is when you have your cell getting depolarized, right? Okay, so the sodium potassium pump maintain the resting membrane potential by pumping out how many sodium? 3 sodium ion outside the cell and 2 potassium ion inside the cell. So that is how you have the resting membrane potential, which is negative 70 millivolt. So we have different type of the channels. First of all, this one is your sodium potassium pump, which requires ATP. Remember, that was the active transport system. And we also have the passive transport system due to the concentration gradient. Like for example, these are some of the what leakage channel, that those are what does not require any energy. Okay, so let's watch this video about the resting membrane potential.
The human brain alone contains about 100 billion nerve cells called neurons. A neuron, like all cells, has negatively and positively charged ions in the fluids located both inside and outside of the cell. However, the ionic composition of these two fluids is markedly different. The inner surface of the membrane is negatively charged, while the outer surface is positively charged. This unequal distribution of charge creates a voltage across the plasma membrane, known as the membrane potential. In a resting, unstimulated neuron, this membrane potential is known specifically as the resting membrane potential. The value of the resting membrane potential depends on two factors. The first is the presence of concentration gradients across the plasma membrane, especially those gradients for sodium and potassium ions. The second is the permeability of the plasma membrane to sodium and potassium. In the resting state, this permeability depends on the number of non-gated leak channels present in the membrane for these ions. The magnitude of the resting membrane potential, inside relative to outside, is measured in millivolts using a voltmeter and 2 electrodes. We'll now place both electrodes in the extracellular fluid. Note that the voltmeter records 0 voltage. This is because the unequal distribution of charge exists only across the plasma membrane. The intra and extracellular fluids are otherwise electrically neutral. However, when we insert one of the electrodes into the intracellular fluid, a voltage is recorded. The value of the resting membrane potential varies, but on average it is minus 70 millivolts.
Okay, so if you look at the different parts of the neuron, they have different type of the gates, different type of the channels in their cell membrane. So if you think about the dendrite and the cell body, these are the receptive. So they have the chemically gated cation channel, chemically gated potassium channel, chemically gated chloride ion channel. And the axon hillock has the voltage gated sodium channel, voltage gated potassium channel. Conductive region also has the same, and transmissive region, they have the voltage gated calcium channel and the calcium pump. So now what did you learn? Which type of membrane protein channel or pump requires cellular energy? Which one? Sodium potassium pump. Which part of a neuron contain chemically gated ion channel? Yeah, very good, because they are the one which is actually receiving the signal. So they should have the chemically gated. Remember that acetylcholine comes and binds with it so that they can generate the potential, action potential. So what type of channels are found along the length of the axon? Voltage gated sodium channel and voltage gated potassium channel. Okay, so now we are going to learn about different ways of the depolarization. Okay, so one way is the graded potential, another one is the action potential. As you can see, the name is already telling you. When we say graded potential, what do we mean by that? So we know that you have inside negative 70 millivolt. Okay. So now if you are changing a little bit, maybe negative 70 to negative 60, that changes, or maybe negative 70 to negative 80. So if you change a little bit, if you have a little bit of change, that is called the what? Graded potential. But when we say action potential, that is a little bit different. So when we say action potential, that means that you have to change the what? This membrane potential from negative 70 millivolt to at least negative 55 millivolt, which is called the threshold. When they reach the threshold, they are like up so much, they are already positive. That is called the depolarization. That is why this is called the action potential. So action potential is all or none. Either you generate the action potential or there is no action potential. Does it make sense? Okay. So now let's talk about the depolarization and hyperpolarization. We all know about the polarization, which is our resting membrane potential. So now what do we mean by the depolarization? One more time. So inside of the membrane become less negative than the what? Resting membrane potential. If negative 70 becomes negative 60, negative 50, negative less, then you call it depolarization. And when we say hyperpolarization, inside of the membrane becomes more negative, and negative 70 is become probably negative 80, negative 90, then you call it hyperpolarization. So now let's talk about this action potential that I just told you, right? So now, for example, think about your brain is sending a signal. When the brain is sending a signal, so what is the signal? So you want to open that sodium channel and all these things, right, for the depolarization. So now when the signal is enough to go at least, to reach at least negative 55 millivolt, which is reaching the what? Threshold. Then what will happen? It can actually reach up to positive 30 millivolt, which is called the depolarization. So as you can see, the unstimulated axon has a resting membrane potential negative 70, but graded these are the graded membrane potential. Graded potential reach the initial segment and are added together. So negative 70 now turn into negative 50. After they reach the negative 50, it's not anymore negative 40 or negative 30. It's gonna reach up to positive 30. So depolarization occurs when the threshold is reached and voltage gated sodium ion channel open and sodium enters rapidly, reversing the polarity from negative to positive. And when we say the repolarization occurs due to the closure of the voltage gated sodium channel and opening of the voltage gated potassium channel, and potassium moves out of the cell and polarity is reversed from the positive to negative. And when we say hyperpolarization, what is happening? Negative 70 will turn into what? More negative, right? So hyperpolarization occurs when the voltage gated potassium channels stay open longer than the time needed to reach the resting membrane potential. During this time, the membrane potential is less than the resting membrane potential. So negative 70 turn into negative 80 millivolt. Then voltage gated potassium channels are closed and the plasma membrane has resumed resting condition activity by the sodium potassium pump. So as you can see in the graph right here, you had the resting membrane potential which was a negative 70. By the graded, if you could reach negative 55, you can actually go up to positive 30, and then what can happen? You can have the hyperpolarization, and finally you will reach the what? Resting membrane potential again. No, it doesn't happen automatically. It's happening all the sodium, potassium. Do you remember we talked about all these things already? How all the sodium and potassium coming out and going out and all these things happening, right? Yeah, yeah. Mm-hmm. Yeah, it's the same thing there. Yeah. So what are the key players? The answer to your question. Why is this happening? Why is this change happening? Why the polarization? Why the depolarization? Why the hyperpolarization? Why it is coming back to the resting membrane potential? Because of these what? So first of all, what is happening? In the beginning, you have the sodium ion channel closed, right? Remember, when acetylcholine come, binds with it, it opens, then sodium enters. When sodium enters, that's when you have the what? Depolarization, right? So you have the open, so when they are open, the depolarization happens. So now when you are trying to what? Hyperpolarize it, what do you have to do? First of all, you have to close that gate again, right? And then what do you have to do? Yeah, so you have to close so the sodium does not enter. And then next, what you have to do? You have to take sodium out of the cell by opening the what? Sodium potassium pump. Remember? So as you can see, what is the first state? Resting state. In the resting state, what is happening? The gate is closed, sodium is outside, so inside is what? Negative. So that is right here, right? Then what is happening? You have the signal to open the gate. When the gate is open, sodium will enter. If sodium enters enough to reach the threshold, it's gonna what? Depolarize to what? Positive 30 millivolts. Then what will happen? The sodium channel will get what? Closed, right? And then potassium will go what? Out. And then what will happen? Hyperpolarization. So now sodium is out, potassium is also going out. So what will happen? Sodium is also going out, potassium is also going out. It's going to be more and more negative. So that is what is the what? Hyperpolarization. Now what do you have to do? Normal condition, resting membrane potential. So repolarization resets the electrical condition, not ionic condition. After repolarization, sodium potassium pump restored the ionic condition. Does it make sense? Yeah. Okay, so now let's talk about the propagation. So when we say propagation, what do we mean by that? So for example, you are sending the signal here. Now you have to send the signal from here to here, from here to here, from here to here, from here to here. That is called the what? Propagation. So now how the propagation happen? Propagation allows action potential to be transmitted down entire axon toward the terminal, right? And sodium influx through the voltage gates in one membrane area cause local currents that cause opening of the sodium voltage gates in the adjacent membrane areas. Once initiated, an action potential is self-propagating. So in non-myelinated axons, if they do not have the myelination, each successive segment of the membrane depolarizes, then repolarizes in wave. That is why that is very, very slow. But the propagation in the myelinated axon differ. That is called the saltatory conduction, because they almost like jump from one node to another node, from one node to another node. That is how it's like really, really quick. Since sodium ion channels closer to the action potential origin are still inactivated, no new action potential is generated there, and action potential only moves in the forward direction. So as you can see how that is happening. So the first is the negative, negative will turn into positive, and this part get positive, and then so it's positive and negative and positive and negative. That's how you are like conducting the signal, right? Okay, so now that is happening, the propagation is happening. So let's watch this video and you're gonna be clear.
Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open.
The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
Okay. So, what did you learn so far? So, it's not only generating the action potential at the one specific site, you have to actually propagate that through the whole axon, right? So that can happen in different ways. The propagation can happen in different ways depending on if you have the myelin sheath present or not present. If you have the myelin sheath present, then you have the what? Nodes of Ranvier. So you have to actually what? Jump from one node to another node to be able to propagate. So that is what is called the saltatory conduction, and that happens very, very quick. If you do not have the what? Myelin sheath, you have to propagate it what? Continuously. First this region, then this region, then this region, like that. So it's gonna be very, very slow and continuous. Okay. So the coding for the stimulus intensity. So one thing I want you to understand right here. So once there is enough signal to generate the action potential, you don't worry about how much signal or how much stimulus is coming. Okay, as long as stimulus is enough to generate the what? Action potential, you will have the full action potential generated. Does it make sense? It doesn't matter like how much. If it is small, if it is very small, too small that it cannot even reach the threshold, that's a different story. But as soon as they reach the threshold, it doesn't matter. It will propagate to its what? Fullest. Okay. So all action potentials are alike and are independent of the stimulus intensity. So how much intensity? It is not a big problem. So central nervous system tells difference between a weak stimulus and a strong one by frequency of the impulses. So frequency is number of impulses received per second, and higher frequency mean stronger stimulus. So for example, as you can see right here, so this is, you are generating some kind of what? Stimulus, but this is not enough to reach the threshold. This is enough to reach the threshold. This is enough to reach the threshold. So as long as they can reach the threshold, they will generate the what? Action potential. It doesn't matter how intense it is. Okay. So as I told you, refractory period. What is the refractory period? One more time. This is the refractory period during the resetting to get adjusted to the what? Resting membrane potential. That is the time your cell is not ready to take another stimulus. That is called the refractory period. So time in which neuron cannot trigger another action potential, voltage gated sodium ion channels are open, so neurons cannot reopen to another stimulus. So there could be two types of the refractory period. One is called the absolute refractory period. So time from the opening of the sodium ion channels until resetting of the channel. Okay. And sure that each action potential is an all or none event and forces one-way transmission of the nerve impulses. And relative refractory period follow absolute refractory period. Most sodium ion channels have returned to their resting state and some potassium channels still open. Repolarization is occurring. Threshold for action potential generation is elevated. Only exceptionally strong stimulus could stimulate an action potential during that time. Okay. So during the relative refractory period, you can still do it, only this is very strongly what? Strong stimulus. Okay. So to reach the positive 30, you have to have enough stimulus to generate the what? Reach the threshold, right? Is that your question? Yeah, resetting. Mhm. That is what is the absolute one, and this is the relative. So relative is the follows the absolute refractory period, that is after the refractory period. So when most sodium channels have already closed, right? Yeah, you were just like at the end, almost everything is done. At the end, you do not still take it, but you will only take it if it is like very, very strong stimulus. Does it make sense? Yeah, yeah. So now your propagation depends on the axon diameter. If you have larger diameter fiber, have faster impulse conduction. The bigger the diameter, the faster the conduction will happen. And degree of myelination. If they have more myelination, more myelin sheath, they will conduct faster. So continuous conduction in non-myelinated axon and saltatory conduction in myelinated axons, impulse jumps from one node to the another node. So some of the homeostatic imbalance. So impaired action potential impulse propagation can be caused by a number of chemical and physical factors. So local anesthetics act by blocking the voltage gated sodium channel. So do you see why you don't feel something? Because of the what? Because of the anesthesia. What they are doing, they are just stopping to generate the what? Action potential by closing the sodium gate, sodium channel. So cold temperature or continuous pressure interrupt blood circulation and delivery of the oxygen to neuron. Cold fingers get numb or foot goes to sleep. So what did you learn so far? What is the repolarization? Positive to negative. Come back to the resting. Very good. What is typically required in order for a cell to reach the threshold? Enough stimulus to reach the negative 55 millivolt. What is the all or none phenomenon? Action potential. When they reach the threshold, they will go up to positive 30, right? How does the distribution of the voltage gated channels differ between the myelinated and unmyelinated axons? So if you have myelinated and unmyelinated axons, what will happen? Yes. So what two factors can increase conduction velocity? If they have myelin sheath present or not, and then the diameter of the axon. Okay. Okay. So now we only talked about one neuron. So it's not only sending the signal through one neuron. You have neuron after another, after another, after another, right? So for example, if you have this one neuron, you have what? Another neuron, right? Right here, right? So you have another neuron. So the, what is it called? This connection between the one neuron to another neuron is called the what? Synapses, right? What do you call it? Synapses. And the neuron before the synapses is called the what? Presynaptic neuron. The neuron, what? This is called the postsynaptic neuron, right? So we're gonna use these terms a lot. So make sure you understand when we say presynaptic neuron, what do we mean by that? And if we say postsynaptic neuron, what do we mean by that? So synapse can be two different types. The synapse we have been talking about so far is actually the electrical signals, sorry, chemical signal, because remember that acetylcholine and then bind with it and then the open, then send the signal, right? So that is the what? Chemical synapse. And there is another type of synapse which is very rare. We call it electrical synapse, less common than the chemical synapse. Neurons are electrically coupled by the gap junctions that connect the cytoplasm of the adjacent neurons, rapid, and found in some brain regions responsible for the eye movement, hippocampus in areas involved in emotions and memory, and embryonic nervous tissue. So as I told you, you have the synapse. When we say synapse, what do we mean by that? This is your axon terminal. This is your next synapse, next neuron. So this is your presynaptic neuron. This is your what? Postsynaptic neuron. So now what do we mean by the axon terminal? We did talk about the axon terminal. So you guys should be very, very good at that, right? When we say axon terminal, what do we mean by that? The end of the axon, right? They are also called the synaptic knob. And inside the synaptic knob, what do you have? All these vesicles. What do you call these vesicles? Synaptic vesicles. And these vesicles are filled with what? Neurotransmitter. SEH is only for the muscle, but for neuron to neuron, it could be so many different ones, right? There are so many different type of the neurotransmitters that we have in our body. Okay. So now axon terminal or presynaptic neuron contain the synaptic vesicle filled with the neurotransmitter, and receptor region on postsynaptic neurons membrane receives the neurotransmitter, and usually on the dendrite or the cell body, and two parts separated by the fluid filled synaptic cleft. That part is called the what? Synaptic cleft. An electrical impulse change to the chemical across the synapse, then back into the electrical. So when we say the neurotransmitter effects are terminated, remember, if you want to stop the action potential, what do you have to do? You have to break down the neurotransmitter or maybe diffuse it out of the place, right? So reuptake by the astrocyte or axon terminal, degradation by the enzymes such as acetylcholine esterase degrade the acetylcholine, and diffusion away from the synaptic cleft. So synaptic delay, time needed for the neurotransmitter to be released, diffuse across the synapse and bind to the receptor, slows transmission to postsynaptic neuron down significantly, but not noticeable because still very fast. What about the postsynaptic potential? So postsynaptic potential is very interesting. So I want you to understand right here. So we can have two different type of the postsynaptic potential. So one could be excitatory, another could be inhibitory. So when we say excitatory, every time we say excitatory, what do we actually mean by that? Depolarization. Very good. So excitatory, signal to depolarize. Inhibitory, signal to hyperpolarize. Okay. Depolarization, excitation, hyperpolarization, inhibition. So you do not want the cell to work, right? Okay. So now this presynaptic neuron is sending the signal to the what? Postsynaptic neuron. So they can either excite them or they can inhibit them, right? So neurotransmitter receptor cause graded potential that vary in strength based on the amount of neurotransmitter released. So time neurotransmitter stays in the cleft. So depending on the effect of the chemical synapse, there are two types of the postsynaptic potential. One is the excitatory postsynaptic potential causes the depolarization, and inhibitory postsynaptic potential causes the hyperpolarization. So as you can see right here, this is the stimulus they are producing, trying to what? Polarize, but did not reach the threshold to generate the action potential, right? But still this is what? Excitatory, because they are still depolarizing, less negative, because they are making it what? Less negative or less negative? Yeah, they are making it less negative. And then inhibitory, what is happening? They are making it what? More negative. They are making it what? More negative. That is why this is called the inhibitory postsynaptic. So now what happens when you have two or three stimulus coming together? Okay. So now for example, you have one stimulus came and it did not reach the threshold. And right after some time, there is another stimulus came and did not reach the what? Threshold, right? What do you call it? So no summation is happening because this stimulation is happening not very close in time. Doesn't make sense? One is coming, going down, another is coming, going down. So they are not enough to actually what? Reach the threshold. So no summation is occurring. However, sometimes you have two stimulus that are coming which are very close in time. So close that they are what? Enough to reach the threshold and generate the what? Action potential. That is called the temporal summation. A rapidly firing presynaptic neuron causes excitatory that are close in time. Okay, when two stimulus is coming which are close in time, they can actually what? Sum each other and enough to generate the what? Action potential. Okay, that is called the temporal summation, and this is the what? No summation. And there is another one which is called the special summation. When we say special summation, what do we mean by that? You are getting the signal from one neuron, another neuron together. You are getting signal from one neuron, another neuron. So you can have more presynaptic neuron. You are getting one signal from one neuron, another signal from another neuron. So that is called the special summation. If more than one presynaptic neuron fires at the same time, what will happen? The generation that different location on the neuron. So that will also be able to what? Do the summation and generate the action potential. So what did we learn so far? So special summation of the inhibitory and the excitatory. When is gonna be inhibitory? So what do you see right here? So this one was excitatory, this one is what? inhibitory. So what will happen eventually? So you have one excitatory, you have one inhibitory. Together, they cancel each other out. Okay. So everyone understand the difference between the graded potential and action potential so far? Yeah. What is the difference between the action potential and the graded potential? Action. Graded potential not necessarily in rest, they're still kind of, yeah, but they are not enough to generate the action potential. It's still kind of imbalance, either less negative or more negative. Okay, I'm not going over all this, but you need to know all the differences between the graded potential and the action potential. Okay. So there are really nice and beautiful charts and comparison. So make sure you know the differences between the, so as you can see, your graded potential, only little bit difference, right? So either what is happening here, either less negative or more negative. These are the what graded potential. And when we say action potential, they are up to what, positive 30 millivolt. Okay, so what are the synaptic vesicle and what are where are they found? Axon terminal. And what are they? They are the small vesicles. What do? They carry neurotransmitter. Okay. How is acetylcholine cleared from the synaptic cleft? You have an enzyme, right, called acetylcholine esterase to break down the acetylcholine into, yeah, muscles, yeah. They will have different type. Acetylcholine can also help in the neuron too, but for specifically for the muscle is the acetylcholine. But we have so many different type of the neurotransmitter. We're going to talk about that too. Yeah. Yeah. In neurons also you have acetylcholine. So which signals depolarization? This one or this one? Excited one? Good job. So what does the word graded mean in the context of receptor potential? Yeah, it's just a little bit of imbalance, right? So you have less negative or more negative in comparison to the what graded potential, sorry, resting membrane potential. So compare temporal and the special summation. When we say temporal and when we say special, what do we mean by that? Temporal is two signal rapid, and special is two signal from the two neuron, right? Yeah. Okay. Now let's talk about all the neurotransmitters that we have in our neurons, right? The most studied one is the what? Acetylcholine, right? That is what we have been talking about a lot. So the acetylcholine is the first identified and best understood released at neuromuscular junction. And there are some other type of the neurotransmitter like the biogenic amines. So for example, catecholamine. Example of the catecholamine is the dopamine. I'm sure everyone is kind of very familiar with the dopamine. It's happy hormone, right? And then the norepinephrine, epinephrine made from the amino acid tyrosine. And then you have the indolamine, all widely used in the brain, plays role in the emotional behavior and biological clock. Imbalances are associated with the mental illness. So there are some serotonin made from the amino acid tryptophan. That is another happy hormone, right? A lot of them are present in our guts to actually secrete the happy hormone serotonin. Then histamine, when does it increase? When you have the allergy, right? Made from the amino acid histidine. Then there are amino acids which could be also neurotransmitter like glutamate, aspartate, glycine, GABA. Okay. And there are some peptide which are also neurotransmitter like substance P, mediator of the pain signals, endorphins, beta endorphins, dynorphins, and enkephalins act as a natural opiate, reduce pain reception. Gut brain peptides, some Somatostatin, cholecystokinin play a role in regulating digestion, and others are purine, like ATP, adenosine gases, lipids, carbon monoxide, hydrogen sulfide gases, endocannabinoids. These are all neurotransmitter. The one of the most important reason why people are interested in drugs is because they want to what, stimulate or destimulate. You can say both, right? Different drugs do different things. Different drugs actually help secreting different type of the neurotransmitter, right? Okay, so a lot of the time I really think about it, and then I think about it like this, like for example, you are so, maybe you had a breakup and you are so sad. Why do you have to take drugs? The breakup is there, right? You are just not feeling that, right? A lot of the time, I am not suggesting you to do it. I do not also want to take medication for a lot of the pains and everything, because the problem is there, I'm just not feeling the problem, right? That is what is happening with the painkiller. Painkiller does not remove the problem, right? Like the drugs does not fix your breakup, right? Exactly like that. So yeah, the way you think about it. So the neurotransmitters are also classified by function. Someone, as I have been telling you, as you are saying, some people take drugs so that they feel it more. Some people take drugs so that they do not want to feel it, right? So some of them are excitatory, some of them are inhibitory, right? Excitatory transmitters causes excitatory, inhibitory causes the inhibitory. And classes by action, some of them work directly. Direct transmitters bind to the receptors that are chemically gated channel. Indirect transmitted bind to the receptors that involve the G protein and second messenger in order to cause the postsynaptic potential. I don't know, you guys remember all this. Do you guys remember the second messenger and all this we talked about during the chapter 3? That insulin receptor, how does that work? Right? Yeah. So you have to know that too. And then the neural integration. This is something important, because the way we have been talking about neuron to neuron, what propagation of the signal, or what conducting the signal from one neuron to another neuron, is not happening linear situation, right? It's not one after another. It could happen so many different ways. It probably a lot of neuron is sending the signal to one neuron, right? Maybe one neuron is sending the signal to so many neurons. Maybe these all neurons are sending the signal to this way, right? We are going to look at some of the picture to be able to understand how how the neuron pulls. So when we say the neuron pulls, functional group of neurons, so single presynaptic fiber branches and synapses with the several neurons in the pull. So there is the discharge zone and there is the facilitated zone neuron. So you have this what, discharge zone and facilitated zone. So you have this presynaptic fiber. So discharge zone closer to the incoming fiber are more likely to generate impulse, so which is your discharge zone. And facilitated zone neurons are periphery of the pool, are further away from the incoming fiber, usually not excited to threshold unless stimulated by another resources. So the one right here is going to be stimulated, but this right here is probably not going to get stimulated. And then the serial processing. So when we say think about the reflex arc, so you always take the information from the environment first by the receptor, and then with the sensory neuron, and then you send the information to the there is the integration center, interneuron, and then motor neuron, and then the muscle, the effector will take the what, action, right? So now the parallel processing, input travels along the several pathways. One stimulus promotes numerous responses, important for higher level mental functioning. Example, a smell may sense smell remind one or of an odor and any associated experiences. Have you ever had that? I have that all the time. A lot of the time some people have, maybe they're using some kind of perfume, and then I smell that somewhere else and I remember that person, right? It's, yeah. Does that happen? Yeah. So as you can see, the circuit can happen so many different. So divergent. So you are sending the information from one through so many together, diverge, right? And there is the converging. So you are sending the information from so many different ways to one, converge, right? And then there is the reverberating. As you can see right here, this one is sending the signal to this line right here. And parallel, so there are so many parallel circuit is coming together to send the signal to this one right here, right? Okay, so what structural category does dopamine fit into? Remember, we talked about some of them are amine, some of them are amino acid, right? Which one is the dopamine? Everyone forgot. Catecholamine. Catecholamine, right? Is the amine. Okay, so the next one, next question. How do indirect neurotransmitter influence their target cell? Yeah, the chemical messenger and the G protein activator. Very good. That's all about this chapter. Does anybody have any question?